Gregor Mendel studied pea plants that were strictly yellow or strictly green, but human traits, like adult height and skin color, present a continuous spectrum. Early geneticists, known as biometricians, argued that these smooth, continuous human traits simply could not be explained by Mendel's discrete particles of inheritance. In 1918, British statistician Ronald Fisher resolved this mathematical standoff. This diagram shows Fisher's solution, known as the infinitesimal model. He proposed that continuous traits are governed by an infinitely large number of discrete Mendelian loci acting together. Looking at the observation bell curve above, you can see the result. The central limit theorem dictates that when you add together the independent effects of all those tiny genetic components, the population naturally forms a smooth, normal distribution. Continuous variation operates entirely within Mendelian genetics. It scales those basic inheritance laws to a level of immense complexity. Polygenic traits lack standard dominance. Capital letters are active alleles, adding metric units. Lowercase are the baseline. Notice genotypes AABBCC and AA capital BBCC. Despite different genetic makeup, they present the exact same intermediate phenotype, because both possessed exactly 3 active uppercase alleles. In a polygenic system, the final phenotype relies entirely on the total mathematical dosage of active alleles, completely independent of the specific letter configuration. This formula breaks down total phenotypic variance, or VP, into genetic variance and environmental variance. Focusing on the genetic components, the additive variance, VA, acts as the true driver for these continuous polygenic traits. We can observe this additive effect in practice. This infographic displays Herman Nielsen Ayle's 1909 wheat experiment, which crossed dark red and white kernels to produce 7 distinct phenotypic classes. Let's run an active recall calculation. If an F2 generation yields exactly 1 extreme white kernel out of 256 plants, how many independent genes control that trait? We use the minimum gene estimation formula. Setting the fraction of the extreme phenotype, 1 over 4 to the power of n, equal to 1 over 256. Solving for n gives us exactly 4 loci. By calculating the mathematical frequency of the most extreme offspring in a population, researchers can accurately reverse engineer the underlying genetic architecture of a trait. Genes do not operate in a vacuum. Look at the right side of this diagram, showing how environmental factors like nutrition or sunlight smear those distinct genetic steps into a smooth, continuous gradient. This chart maps that blurring effect, defined formally as phenotypic plasticity. A single genotype maintains the capacity to produce a wide range of phenotypes, reacting directly to external triggers like temperature and UV light. To isolate these minor effect polygenes, geneticists relied on mapping techniques. Here, illuminated on the chromosome, are quantitative trait loci identified using statistical association studies. This table provides a complete comparative framework, contrasting qualitative Mendelian counts directly against quantitative polygenic statistics and bell curves. Keep this handy. By combining discrete DNA tracking with environmental data and statistical variance, we have the exact mathematical blueprint required to map biology's most complex traits.